of the world to New Zealand to the Malaga Institute of Medical Research to perform his postdoctoral researcher studies at Franca Ronchese lab. And then since 8 months ago, he started his own lab at the Philips University in Germany. And congratulations on your work, and we are happy to see you here.
Yeah, thank you so much for the introduction and also for the opportunity to present at Open Box Science. It's my great pleasure to present my work to you today. And I think I speak for the whole audience that we as immunologists love to subset the immune system and define novel cell populations, an exercise which I think has really exploded in recent years thanks to single cell technologies. And so it has really led us to the important question of how we define an actual cell subset or a cellular phenotype, and where we draw the line between a phenotypic observation or a functional change in this population. And a very relevant classification which has withstood the test of time is based on cell ontogeny. And in the context of antigen-presenting cells, which are the cell population that I study a lot, we know that lineage and developmentally speaking, there are 3 types of antigen-presenting cells. The one type are the conventional dendritic cells, which develop in the bone marrow in all mammals from hematopoietic stem cells under the control of FLT3 ligand, and then divide into different lineages of conventional dendritic cells. You have a second group of monocyte-derived cells, which also derive from bone marrow hematopoietic stem cells and are under the control of MCSF or GM-CSF. And you have a third lineage, which is related to this monocyte macrophage lineage, at least in phenotype, but actually looking at the ontogeny is derived from embryonic precursors within each tissue, which then divide and self-replenish within tissues even in adult life. And so based on this classification on the ontogeny of antigen-presenting cells, we can then clearly look in each tissue or even in the circulation, and we can define circulating cells that have come from the bone marrow as progenitors, which then seed all tissues or lymph nodes. And we can look at the populations that are resident and that divide locally and expand locally. But if we take this division and now use our single cell, either flow cytometric analyses or single cell sequencing analyses, we come to the observation that one population of conventional dendritic cells, the type 2 dendritic cells, stands out from this because we and many other labs have observed that this population of dendritic cells really expresses a tissue-specific pattern of different surface molecules, also of transcriptomic signature, and has the potential to induce different populations of dendritic cells depending on the experimental setup. So to explain this subgroup of dendritic cells and how this observation might match a biological phenomenon, 2 hypotheses have been generated. The first hypothesis suggesting that type 2 dendritic cells form a homogeneous group, which is stimulated or conditioned by different antigens, and that the properties of these antigens lead to the specific phenotypes we can observe and to the functional regulation of different types of T helper cells and T helper cell differentiation. The second hypothesis actually suggests that within this previously believed homogeneous subset of type 2 dendritic cells, there are actually further subtypes that need to be identified, and that each of these subtypes has the molecular machinery to process and present unique antigens, which then lead to a unique functional immune response later on. And so we try to address these 2 hypotheses and try to use novel tools at the Malaga Institute to really look at this population of type 2 dendritic cells. And so in the first set of experiments, we used high-dimensional flow cytometry, which included all surface markers that have been reported for this population. for flow cytometry. And we basically could see when we now pooled dendritic cells from the lymph nodes from different tissues, we could see that very clearly these three ontogenetic lineages of dendritic cells, the type 1, type 2 dendritic cell, and the Langerhans cells clearly segregated in an unbiased manner. And when we then looked into the type 2 dendritic cells, these population or cluster of cells uniquely furthermore divided by their origin. So if they derive from the skin, the lung, or the intestine, which suggested that the tissue had a unique impact on the surface marker expression of these cells. And when we used deconvolution strategies to actually define unique markers that were tissue specifically expressed by these type 2 dendritic cells, we found that in the skin, the expression of CD11b led to a unique profile. That one in the skin, you could find CD11b high type 2 dendritic cells, but also CD11b low type 2 dendritic cells. In the lung, there was a unique expression of CD24 at the steady state. And in the intestine, there was a unique expression of CD103. We then wondered if this had to do with migration or if the lymph node observation we made was also matched by the expression of these markers within each tissue. And so we looked at each of these individual tissues and the draining lymph nodes. And again, we could observe that these populations were present at a constant rate, suggesting that they develop in accordance to tissue-specific signals and then migrate at a constant rate at the steady state to the draining lymph nodes. We also looked into male and female mice, so this was on a black 6 background, and we observed that although there were small changes observed in the skin, overall these populations were also observed independent of the sex of the mice. And in addition, we looked at different backgrounds of mice, so C57 and BALB/c genotypes, and we observed that also there was a constant pool of cells that had these tissue-specific phenotypes. Because the tissue phenotype of a few markers that we detected by flow cytometry is not really enough to define if they are a cell population and if they really have a unique transcriptomic profile, we sorted these different populations from these different tissue draining lymph nodes and performed bulk RNA sequencing. And we observed that although we put in 6 different populations from 3 different tissues, when we looked at the transcriptomic profile and the transcriptomic clustering, we could actually identify 4 different populations because 2 populations actually grouped together, whereas 2 populations set apart based on their transcriptomic signature. And the populations that really expressed a unique transcriptomic signature were the CD103 positive DC2s from the intestine and the CD11 below DC2s from the skin. Now, previous work actually from a neighboring lab in Scotland, where I did my PhD under the leadership of Alan Mowat, actually had already studied the CD103 positive DC2s within the intestine and found through elegant mixed chimeric experiments that this population of CD103 positive CD11b double positive DC2s within the intestine was dependent on TGF-beta receptor signaling for their development, and that in the absence of this population, you could observe mild colitis irrespective of the T regulatory cells, clearly already defining that local signals such as TGF-beta signaling plays an important role for the development of this population in the intestine. And so we wanted to ask, because they had not observed that the same signaling plays a role in the skin, we now wanted to identify what sort of signals play a role in the development or functional specification of these CD11 below DC2s from the skin. And so we performed upstream analysis on the transcription factor binding sites, focusing on these unique gene expression profiles of each of the clusters we had identified by RNA sequencing. And we observed to our surprise that the cluster 3b, which represents CD11 below type 2 dendritic cells from the skin, had a unique expression or enrichment of several transcription factors, which are listed here, which based on the literature search have several properties in the involvement of T cell interaction and antigen presentation, but also we found a strong enrichment for the transcription factor STAT6, which really puzzled us because STAT6 is strongly involved in type 2 immune responses, but has limited involvement in steady state immunity. And so we were really surprised to find the enrichment of this transcription factor in this very unique population of dendritic cells, which was not present in other or not enriched in other dendritic cell populations, ruling out that these mice were infected by parasites in the mouse facility, for example, because then one would expect an enrichment in all populations, or at least in all populations in the skin, which was not the case. So we then tried to experimentally address this, and we looked at STAT6 knockout mice, which were also on a C57 Black 6 background. And what we observed was that when we looked specifically in the skin, we saw a complete reduction of this CD11 below DC2 population, whereas when we looked at the lung or the small intestine, there was no effect of STAT6 signaling or the absence of STAT6 signaling, which really showed that this enrichment that we found transcriptomically really has a strong effect on this unique population of disease and not on other populations that are also present in the skin. We then wondered if our observation by flow cytometry and on the expression of CD11b was an effect that was directly regulated by STAT6 or was only regulating CD11b expression, but not the population as a whole. And so we performed single cell RNA sequencing and again looked at the type 2 dendritic cells of Black 6 control mice or STAT6 knockout mice. And what we looked in an unbiased fashion at this population of CD11 below DC2s, which you can see here in purple, we could indeed confirm by single cell sequencing that they are absent in this STAT6 knockout mice, and that it is not only the expression of CD11b that is regulated by STAT6, but indeed the development or differentiation of this population as a whole. As STAT6 is activated through IL-4 and or IL-13 signaling, we next wanted to understand the upstream regulators of STAT6. And so we looked at our type 2 dendritic cells in IL-4 knockout or IL-13 knockout mice, and we observed that at the steady state, IL-13 knockout mice showed a deficiency for this population, but IL-4 knockout mice did not, indicating that at the steady state, IL-13 activates STAT6 signaling and leads to the differentiation of this cell population, whereas IL-4 is not relevant at the steady state context. Indeed, we could also confirm that IL-13 signaling was mediated through the conventional IL-13 receptor, which is formed by IL-4 receptor alpha and the heterodimer with IL-13 RA1. And we could see that in the knockout mice for each of these components of the IL-13 receptor, there is defective downstream signaling, no phosphorylation and activation of STAT6, and no development of the CD11 below these twos in the skin. We, of course, then wondered which cells produce IL-13 at steady state, because upon looking at the literature, there was very few reports and no functional reports of IL-13 being expressed in the skin of Black 6 mice or mice in general at the steady state. And so again, we used high-dimensional flow cytometry on IL-13 reporter mice to look at the immune cell compartment of the skin and identify which cell at the steady state is expressing and producing IL-13. And at the steady state, it was very clear that IL-Cs were the producers of IL-13 in the skin, and that skin ILCs were uniquely producing this IL-13 at the steady state, whereas ILCs from the lung or from the intestine from those same animals did not express IL-13 at the steady state. When we phenotyped those IL-13 positive ILCs in the skin, we could further identify that they were not producing IL-4, but only IL-13, and that although they expressed IL-13 and would thus belong to the ILC2 lineage, we could not find the expression of classical ILC2 molecules such as IL-33 or CD25 or KLRG1, which really was perplexing to us because the phenotype of those ILCs, albeit the expression of IL-13 had a resting phenotype and was only characterized by the expression of CD44 and ICOS. We then wanted to identify if IL-13 was not only necessary for the development of these CD11 below DC2s, but was also sufficient to drive this development. And we performed two rescue experiments. The first one was in vivo, where we injected IL-13 fusion protein into IL-13 knockout mice, and we could observe that even after only 4 days of injection of this fusion protein, we got a complete rescue of this CD11 below DC2 population. And that in vitro, when we added recombinant IL-13 into bone marrow dendritic cell cultures, we could also observe the development of a CD11 below DC2 population, which we could also confirm by qPCR, had similar expression to the in vivo counterparts, clearly indicating that IL-13 is not only necessary in the skin to drive the development of this CD11 below DC2 population, but is also sufficient to drive this phenotype. Next, we wanted to understand, now that we knew the developmental pathway and requirements for the development of this population of dendritic cells, we now wanted to understand if they were merely a phenotypic phenomenon, observation, or if they had functional properties as well that made them a unique functional population of type 2 dendritic cells. And so we developed two in vivo mouse models where we observed, or we generated, a lack of CD11 below DC2s that was independent of a T cell response or a T cell defect. And the first one was a 50-50 chimera, where we could interrogate T cell responses which were not impaired STAT6 signaling, and where we had antigen presentation which either was normal and undergoing normal development of CD11 below DC2s, or in the second group of mice had a deficient development of CD11 below DC2s. We then immunized these chimeras with different antigens, which either drove a Th1 response in the case of Mycobacterium smegmatis, a Th2 response in the case of Nipostrongylus brasiliensis, or a Th17 response in response to Candida albicans. And what we observed was that in these chimeras and during immunization, it was irrelevant for the existence of the CD11 below DC2 population or the absence of the CD11 below population in regards to a Th1 response. So there was no difference between the two groups of mice. However, when we immunized these mice with a type 2 inducing antigen, so for example here a parasite-derived antigen, but we had also looked at allergens, we could observe that in the group which did not have this CD11 below DC2 population, there was a very suboptimal response in regards to type 2 and Th2 immune responses. Whereas when we immunized those mice that did not have CD11 below DC2s with yeast or fungal extracts and fungal antigens, we actually observed that there was an increased Th17 response in this scenario. We confirmed this observation using a conditional knockout mice system, where alpha receptor alpha expression was under the control of a dendritic cell specific promoter, which again led to the very specific depletion of those CD11 below DC2s without affecting any T cells in this signaling. And again, we observed that Th1 responses were not affected, but that in the absence of CD11 below DC2s, there was a strong reduction in the Th2 response and respectively a strong increase in the Th17 response. Going beyond the mouse observations, we also asked ourselves if similar populations of dendritic cells would also exist in the skin of humans. And so we looked and data mined several publications which had looked at healthy individuals and had phenotyped dendritic cells within the skin or other tissues from the same patient. And what we could observe was that genes linked to the IL-4 and IL-13 signaling pathway in human dendritic cells were enriched in the skin, but not enriched in other tissues such as the blood, the spleen, and the lung. And so clearly indicating that IL-4 or IL-13 signaling also plays a role in the skin and also directly affects dendritic cells within the skin in healthy individuals. And we could confirm this in another data set which had looked at dendritic cells on a single cell level, and we could also observe that, for example, IL-4 receptor expression, IL-13 RA1 expression, and also STAT6 were enriched in those dendritic cells from healthy individuals within the skin, and that also IL-4 and IL-13 expression was observed in healthy skin in both T cells and ILCs at the steady state, suggesting that this signaling could have a direct effect on the dendritic cells and might lead to a similar differentiation and specification of type 2 dendritic cells in the skin. And this is basically to summarize our finding that we found that homeostatic levels of IL-13 exist in healthy skin, and that in mice at least, the predominant producers of IL-13 are innate lymphoid cells, which have a resting phenotype, and that this IL-13 has a direct immunological function in naive skin because it influences the development and differentiation of type 2 dendritic cells precursors and drives the development of this CD11 below DC2 population, which then plays an important role in controlling the Th2 and Th17 balance in healthy skin. And when any of these pathways are blocked, you get a strong reduction in the CD11 below DC2 development, which then leads to reduced Th2 responses and an increased Th17 immunity and a dysregulated balance within the skin. And so we could clarify that at least in the example of the skin, there is a default antigen-dependent conditioning of type 2 dendritic cells because Th2 responses in our mouse models were not completely abolished. So there is some sort of capacity for the remaining type 2 dendritic cells to deal and drive Th2 responses in a default context. However, we also found that in the context of the skin, there is a unique population of type 2 dendritic cells, the CD11 below DC2s, which perform a very subset-defined function, and in this case drive specifically Th2 immunity and control Th17 responses. And there might be an evolutionary origin or necessity to develop additional populations of dendritic cells in specific tissues. And we are now in my new lab trying to understand what the requirement, evolutionary requirement for this additional population of dendritic cell is. And if it is, for example, to promote wound healing or to defend against toxins or parasites at the steady state, which is why this population has developed over time. But also, if we're now looking at more modern diseases, for example, allergy or autoimmunity, are these type of dendritic cells in the skin uniquely predisposed and uniquely contributing to these types of diseases where wound healing or parasite infection might no longer be required? And so we are now working on this in my new lab. And I just want to also introduce my new website that, you can find different description of these projects and also different opportunities to join the lab. And so for the end, I want to thank my supervisor, Franca Ronchese, and also Olivier Lamiable for their great contribution to this work, and the entire group at the Malagan Institute, where I did my postdoc, and where we worked on this project. And I thank you for listening, and I'm happy to take questions.
Thank you very much for this great talk. So I think, yes, we have time for more questions. So the first one is from Miranda. So she asks, during infection or immunization, do other cytokines override homeostatic IL-13 signaling? Is STAT6 further activated in these settings?
Yes, so that's something that we now want to look at. For example, if we have a type 2 immune response and we have the production of IL-4, does IL-4 augment the differentiation of CD11 below DC2s and also play a role in their differentiation? Or if we now have activated ILC2s, that not the resting one, but activated ILCs in the skin that also produce high levels of IL-13, does this also play a role in dendritic cell differentiation, or is it a unique location situation that might not affect this directly?
Okay. She also asked if IL-13 is regulating, is it specific only for this dendritic cell, or actually it could be regulated or other type of dendritic cells, including CD301B dendritic cells?
Yeah. So when we actually looked at our high dimensional phenotype, the panel also had included CD301B. And what we found was that CD301B was not segregating unique tissue-specific populations, but was a phenotype of all type 2 dendritic cells. So all type 2 dendritic cells had different levels of expression, so had a positive population of CD301B and also a negative population of CD301B. And actually, when we looked at our STAT6 knockout or IL-13 knockout mice and gated this time on CD301B positive dendritic cells, we could not observe a difference in this population because they are not specific, at least in the skin, for CD11B high and CD11 below DC2s, but they are actually expressed equally by both of these subsets. So they are not regulating this.
Okay, so Damian Maceda asked if, great talk, in some skin autoimmune diseases, there is a preference for Th2 overactivation. Do you think some of your models be useful to avoid Th2 responses?
Yeah, so actually here in Marburg, there is a lot of study on pemphigus disease and Bolus skin diseases. And there's actually, as you mentioned, a very strong bias towards Th2 responses in these autoimmune diseases. And that's actually why we are currently hypothesizing that it is the CD11 below DC2s that are specific to the skin that actually drive this type 2 flavor of the disease. Whereas if we, for example, had an autoimmune reaction in another tissue where those cells do not exist, we would not see this Th2 bias. And that's currently something that we're trying to model in vitro, and then also hopefully we'll be able to take into in vivo experiments soon.
In fact, I would like to ask a question. What is the signal that drives IL-13 production in the skin on the IL-6?
Yeah, that's a very good question. That's also a grant that I'm about to write or about to submit. And that's exactly asking this question because in the paper, there are further experiments that we did looking at germ-free mice, looking at IL-33 deficient mice, IL-25 deficient mice. So basically taking all the signals that we currently know from the literature that should activate ILC2s to drive IL-13 production did not have an effect on this homeostatic IL-13 signaling. So these ILCs that are producing IL-13 are not the conventional ILC2, are not regulated by microbial signals or by IL-33 or by IL-25, but are regulated by something that we don't know yet. And that's something that we think is very exciting because it really brings also a new flavor, not also to the field of dendritic cells or T cell regulation, but also into the field of ILCs.
Thank you. If there's not any more questions, I would like to thank both of our speakers today. And just to remind, follow our Twitter account on Facebook as well. You can follow for our upcoming symposiums. Actually, we have some on the IL-13 actually, and where is the post? Yes, interleukin-13 and its receptor are synaptic proteins and involved in plasticity and neuroprotection. We're looking for that next meeting. Well, thank you very much, and see you next time. Bye bye. Thank you.
Thank you all.
Thank you.
Thank you. All right.